Good afternoon and welcome back to Virology. So this week we're going to look at two kinds of virus infections. Today we're going to talk about acute infections, and Wednesday we'll talk about persistent infections. Now what's the difference? Here are some graphs of acute and different kinds of persistent infections. And what we're looking at is virus production on the y-axis and time on the x-axis. And so the top one is an acute infection where the disease is shown in red. It's a defined period of disease. It doesn't have to be short, it's just defined. And also a defined period of virus shedding, which is shown in the blue curve. Rhinovirus, rotavirus, influenza virus, SARS-CoV-2, many poliovirus, many examples of acute viral infections. The others are persistent. By definition, a persistent virus infection remains with you your whole life. You can't get rid of it. The only way is when you die. You're done. And we'll talk about those next time. So acute infections, rapid onset of virus production, limited but not necessarily short course of disease, can be severe, could be longer, doesn't have to be just a few days. Production of large numbers of virus particles, and eventually immune clearance. Those are the characteristics of acute infections. So here's a time course of a typical acute infection. Again, rapid and self-limiting is the key here. And virus production on the Y, time on the X. So we have the infection beginning there at the origin, and the red is virus reproduction. So you can see virus begins to reproduce. The gray is the threshold of virus needed to activate the adaptive immune responses. So initially, the innate defenses are at play from the beginning of infection to the first hours. And most infections are in fact controlled by the innate defenses. But many are not controlled, so the titer exceeds this threshold, and then we have activation of adaptive responses. But typically the peak of virus reproduction occurs before adaptive responses kick in, and in fact, they play little role in clearing virus. It's more likely that the innate response plays a big role in how far it will go. And then, of course, the virus is cleared or controlled, and then we have memory, immune memory, as we talked about. An important feature of acute infections are inapparent acute infections. These are successful infections with no symptoms, right? No symptoms. You don't feel anything. That's what an inapparent infection is. You do have signs, right? The virus is in you, it's multiplying, immune response is kicking in. So those are signs of infection, but there's no symptoms. And you can make enough virus particles to spread in the population. So asymptomatic infections are a very important contribution to transmission of infection. Now, how do we know that there are so many asymptomatic infections if there are no symptoms? Well, we do serological surveys. We take serum from a number of people throughout a country, and we look for antibodies to the virus. And when we take the serum, we ask them, have you had these symptoms? And we can find a lot of people who are seropositive, they have antibodies to the virus, but have had no symptoms. And so those are asymptomatic people. And viruses with high asymptomatic rates are generally what we consider well-adapted pathogens. They've been with their host for many years, but another example is poliovirus. Over 90% of poliovirus infections are inapparent. So the inapparent infection is a really key aspect of an acute virus infection. Now, these are typically major public health problems, which is not to say that persistent infections are not. But these are as well. They cause serious epidemics, affecting millions of people each year. Influenza virus, which we'll talk about today, Norovirus, and many others. Millions and millions of people. And many deaths. Although most people do recover, it has public health costs. The problem with an acute infection is by the time you have symptoms, the infection may be over, and it may already have spread to someone else. We'll see some examples of that today. And that's what makes it challenging to control acute infections, and why really the best way is vaccination, as we'll see later. First question is, which of the following do acute infections and incubation periods have in common? The virus is not replicating, no symptoms are visible, immune defenses are engaged, the immune system does not respond, all of the above. So which are in common between acute infections and incubation periods? Okay, let's look to see how we did. So the answer is C, immune defenses are engaged. These are common between acute and incubation period. So the virus is not replicating. Obviously, it replicates in an acute infection and during the incubation period. No symptoms are visible. That's the incubation period, but not the entire acute infection, right? Immune response doesn't respond. Yes, it is responding during both. And so the answer is C. All right, so I'm going to talk today about 5 viruses that cause acute infections. And I picked them because 2 of them are transmitted by the respiratory route, influenza virus and Measles virus. 2 of them are transmitted by fecal-oral route, poliovirus and norovirus. And 1 is transmitted by mosquitoes, West Nile virus. So let's start with influenza virus. There are 3 types. Remember, this is an envelope negative strand segmented RNA virus. 3 types, A, B, and C. A and B cause a rather similar disease. We vaccinate against A and B. C infects a lot of people, but mostly inapparent illness, and we don't vaccinate against C at all. And only A causes pandemic. Pandemic is a global outbreak, which can happen every 10, 20, or 30 years for influenza virus. We'll see later. And the hallmark of influenza viruses is antigenic variation. We've talked about this before. Now, the transmission of influenza virus occurs in a couple of different ways. First, by droplets produced by coughing or sneezing or talking. You saw this picture before. We make droplets of different sizes that go different distances in the air. So that's a major route. But also contact. If you shake someone's hand and they've recently touched their nose and their mucus, you could get contamination with virus, or contact with contaminated surfaces. So you touch a doorknob that's heavily contaminated. Someone has just opened the door who has flu and they have mucus on their hands. They get it on the doorknob. You pick it up and then you touch your nose or eye and you transfer it to yourself. So my advice has always been, if you're in public places, you obviously have to open the door, but make sure you wash your hands before you touch your eyes or mouth. I'm still using gloves here. Even though it's warm, I'm carrying gloves and I use those to open things. Maybe I'm a little paranoid. So the virus enters the upper respiratory tract. You inhale it typically, or you can pull it in. Some people are mouth breathers, right? You can pull it in through your mouth as well. And it reproduces in the upper respiratory tract in the epithelial cell layers. And then very typically, it spreads to the trachea. And that's why if you have influenza, you often have a substernal pain associated with it. That's virus reproducing in the tracheal epithelial cells. In the most extreme cases, it goes down into the alveoli and can cause pneumonia. This is a very serious illness. Now, the receptors control where influenza viruses can infect. Now remember, human influenza viruses bind to alpha-2,6 sialic acids as a receptor, whereas avian bind to alpha-2,3. Now in our upper tract, we have mainly alpha-2,6. You can see nasal mucosa, paranasal sinuses, the pharynx, the larynx. It's only when you get to the trachea do you start to find alpha-2,3, and then you found alpha-2,3 in the bronchi, the bronchioles, and the alveoli as well. And so you have to, you're inhaling the virus and getting infected in the upper tract. So avian viruses have a hard time doing that. It's usually human viruses that will start an infection. There's one exception though. Well, if you are people who live with chickens, so in many countries of the world, people breed chickens and they actually sleep with them. And so you're inhaling deeply all night, and that can get down into your lung. Also, there's alpha-2,3 receptors in the eye here. You can see alpha-2,3 in many places. So you can get an eye infection, and then the virus can drain through the duct into your upper respiratory tract. And so if you've been listening, you know, in the US, avian H5N1 virus has infected cows, dairy cows. And you know, it causes mastitis in them, so that they don't get very sick. But it can transmit to dairy workers. We've had a number of infection of dairy workers. It typically is associated with conjunctivitis, because that's where the virus can first replicate, the alpha-2,3 receptors. And then that's all that those individuals get. They really don't get severe lung infections. So the receptor can control what kinds of viruses you get. So uncomplicated influenza, 1 to 5 day incubation. You know, it depends on factors in each host. Very quick onset. You can tell the hour that you got influenza symptoms: headache, chills, dry cough, high fever, myalgias, malaise, anorexia. The fever peaks within 24 hours, 38 to 40 C. It's very high. And then it declines and is gone by day 6. But then as you're not done when the fever declines, respiratory symptoms intensify as the fever declines, and you start to have a productive cough, which means it's full of mucus, right? You can, and when you cough, you can hear that mucus bubbling in you. And that can last for 1 to 2 weeks, all right? So it's an extended infection. You feel pretty miserable after influenza. How do we diagnose it? The first is by what we call influenza-like illness. Fever of at least 100 degrees, cough or sore throat, and no other known cause. We now have a flu-COVID rapid antigen test, because those are two very common viruses to find in the respiratory season. And we can use that to know what you have there. And there are rapid antigen tests, but they're not great. They have a lot of false negative and positive results. The best is PCR, which takes more time, or viral culture and serology. So if you go to a healthcare here at the university, they will see you have fever, cough or sore throat. They'll just give you a prescription for a flu antiviral. Otherwise, they could do a rapid antigen test. So here are some challenge experiments which you can do with influenza virus. Certain strains are very mild. And what we do is we put influenza virus in the nose, and then we look at days after infection. We measure viral titers, so you can swab and measure titers. And you can also ask people about symptoms. So you can ask them certain questions and give it a score. So here the virus titer. You can see already in 1 day, you have quite a bit, 2 logs of virus, peaks at 2 days, very quick, and then starts to go down. And the symptoms are a little bit lagging behind. So I always say about 2 to 3 days you could shed influenza virus before you get symptoms and transmit it to other people. On the right is another experiment of a challenge, days after administration, and we're looking at either virus titer in the nasopharyngeal wash or our antibody titer here in the wash or serum. And you can see virus, again, peaking very quickly and going down very quickly, a rapid peak of interferon. And here are the antibody titers. I mean, they're coming up when the infection, the reproduction is almost over. The symptoms are not over. You're going to feel crappy for 2 weeks, but it's coming up late. And so I think the main key of clearance is interferon for influenza. Now, influenza is a seasonal disease. This is a chart of seasonal influenza from 2013 to 2022. And these are the winter months. It's not shown here, but it's peaking in the US. This is US in the winter months. And the different colors are the different A and B influenza viruses. So every winter you get a peak, and the peaks are different in intensity. This is the number of specimens that are testing positive on the y-axis. And notice there are no data for 2021 because we were all locked up. And so nobody transmitted influenza virus. So if you want to stop transmitting viruses, just stay home all the time. Seasonal influenza. Influenza can kill you. So typically, this is the data from 2010 and 2023 from CDC. So we've had between 9 million and 41 million illnesses. So that's on a yearly basis. We've had between 100,000 and 700,000 illnesses, depending on the year, and between 5,000 and 51,000 deaths. This is just in the US, and that's from year to year. And so here are the data from year to year. You can see the gray is the symptomatic illness, which varies. The yellow is hospitalization, and then deaths. And you can't really see the deaths here, but the number of deaths vary between 5,000 and 50,000 a year. So there's a lot of people dying. So don't think flu is nothing, right? It's killing. And those hospitalizations, when you go in the hospital for a respiratory illness, not good. You can end up getting intubated with oxygen, and that's not good. Intubation really messes with you. The US does lots of surveillance for flu. In fact, many countries do. Here is this season's influenza. So every week, the CDC reports. This is influenza positive tests reported by US public health laboratories. And it's divided into the different kinds of influenza viruses. So here, yellow is A, and this one is H1N1A, and red is H3N2, and then the B is the green down here. So it's a fraction of the A. So the A influenza viruses, right now there are two subtypes circulating, H1N1 and H3N2, and they vary from year to year. In this season, H1N1 was predominant. And that's the virus that arose in 2009 in the pandemic that happened back then. But you can see we are now past the peak. The peak here was week 6, so that's like mid-February, right? And so now we're in week 10, January, February, the first 2 weeks of March here. So we're steadily going down. And by May, there'll be very, very little influenza activity like you see over here. So this is week 40 here in 2024, very low activity. So starts coming up roughly week 50, the end of the year, right? Okay, so that's one statistic I want to show you. And then there are many others you can find on the CDC website. Every day you can go and look at influenza activity in the US. They color each state a different color depending on the activity. So this was in March. Activity is going down now. So a lot of these states are green, right, minimal. But some of them are still red, high activity. You can also see on the left here mortality, influenza mortality. And this is weak. So you have your seasonal peaks of mortality. And look, this is this year. This year was a serious year in terms of influenza mortality. We had a lot more than we have had in recent years. And then on the right, this is the percent of outpatient visits for respiratory illness, another measure of how pathogenic or how virulent the virus is. So the 24-25 season are the red dots here. This again by week, and you can see pretty high outpatient visits. This is the number of visits for influenza-like illness. And in some seasons, it's much lower, like the purple one or some of the others. But this one was very high. And then, well, I guess this is the same one, yeah, that I put in twice. So you can see it a little better. This is the outpatient visit for 2025, which is equaling the 2019-2020 season. And you can see this one, the 2018, was pretty mild, as was this season as well. So there are complications of influenza. I told you about this 2-week course, but there are complications that can make it much worse. You can get pneumonia. The virus goes down into the alveoli and starts to reproduce in those cells and destroys them. You have trouble breathing. It's primary viral pneumonia. You can also get secondary bacterial pneumonia. After you start to recover from influenza, the bacteria start to multiply and you get like streptococcal pneumonia. Muscle pain, you can have cardiac involvement. So some people have so much trouble breathing, their heart is overworking and you can have heart issues. And then you can have this syndrome called Rye syndrome, Rye, which is encephalopathy and liver damage. What can you do about it? A lot of things you could do. You could do non-pharmaceutical stuff, you know, like you go to the drugstore, you buy all those lozenges and liquids and so forth. There's a lot of those. But there are antiviral drugs. There are 2 neuraminidase inhibitors. There is an inhibitor of the ion channel in the virus membrane, and there's an inhibitor of the endonuclease. So we'll talk more about those when we talk about antivirals. You have to take those within a day or 2 of testing positive, otherwise they will do nothing. So if you feel crappy and you go get tested and you're positive, get it right away, otherwise it won't help. And then we have vaccines, of course, and we'll talk about those later as well. Which of the following is characteristic of uncomplicated influenza? Transmission may occur via respiratory droplets. The incubation period, 1 to 5 days. Fever peaks in 24 hours. Coughing and weakness can last 2 weeks. All of the above. Okay, let's see how we did. All of the above, that's right. Almost 100. They're all characteristic of uncomplicated influenza. All right, let's go to measles. It's a member of the paramyxovirus, a negative stranded envelope virus, a very contagious human virus. It's estimated that an infected individual could, on average, infect 15 other people. If I had measles and I were standing up here talking, you would all get measles. It's that transmissible. But I don't. I had it as a kid. I'm good. There's one serotype of measles. It doesn't change. And infection confers lifelong protection, transmitted by respiratory droplets like influenza virus. And here's the key for measles. The incubation period is 2 weeks. But you are shedding virus 2 to 3 days before you get that characteristic red rash, which says, aha, you have measles. So you can be shedding virus for a couple of days. You're in school even. And almost everyone shows symptoms of diseases. There's no asymptomatic infection. Signs and symptoms, it should be, because if you're going to show symptoms, you're going to go show signs. So I'm going to change this right now. Okay, so here's the pathway of the virus spreading in you. You inhale it. You inhale virus, goes to your respiratory epithelium. Does not infect the mucosal epithelial cells. Rather, it infects immune cells that are throughout the epithelium, like dendritic cells and macrophages. Remember, they're sentinels. They bring the virus to the lymph node, and it reproduces in lymphocytes, which the lymph node is full of. So now the lymph nodes produce a ton of virus, which goes to the blood, and you have your primary viremia. And that brings the virus to multiple organs, including the skin, to produce that rash, and in some children, the central nervous system as well. Okay, so now you have this rash, which by the way is contagious. There is virus in the rash, and the virus is flowing through your blood vessels. At some point, that virus in the blood reaches the basolateral surface of the respiratory mucosal epithelium, right? So the virus is released from lymphoid cells on the basolateral surface. It binds to a receptor called Nectin-4 and replicates in these cells and is shed at the apical surface. And then that is how it spreads to other people. So think about this. This is very interesting. When it infects you, it doesn't infect the respiratory epithelium. There are no receptors on the apical side. Instead, it's picked up by immune cells. Viremia is established. That brings the virus back to the basolateral surface of the respiratory epithelium. The virus is reproduced there and are shed. That's how it spreads to other people. Uncomplicated measles, high fever, respiratory systems, runny nose, cough, conjunctivitis, and the rash from face to extremities. It starts at the face, so the rash initially begins in the face, and then it spreads downwards. So if you catch it early, you can see that. And you get rash over the whole body, as you can see this kid. And in the mouth, if you look, are what are called Koplik spots. And these are little white spots here. You can barely see them, but they are a result of the measles virus infecting the epithelial cells inside your mouth and causing syncytium formation. Remember syncytia? Fusing of cells so that you have one cell with many nuclei. These Koplik spots are syncytia. So in the old days, you could look at a kid who had a rash, Koplik spots, that's it, got measles. All right, on the bottom here is a timeline, very important. Infection here at day 0, incubation period, no symptoms. Then we have a prodromal period where you have Koplik spots, fever, malaise. And so, depending on how you feel, you may go to school because you're shedding during this period. And then you have the rash at day 14, and you continue to shed, and then eventually recovery. So you can be around other people for quite a while and shed virus without being symptomatic, and that's what happens in these outbreaks. That's what happens in Texas. Kid gets measles. It's okay, he's shedding virus, goes, plays with other kids, the other kids get measles too. Because they're all unvaccinated, of course. You have lots of complications with measles. Encephalitis, 1 in 1,000 kids, bronchitis, pneumonia, ear infection. Many kids end up deaf after measles virus infection. Fatality. In the US, 1 or 2 kids per 1,000 die of measles. This number is 30% in countries with poor nutrition. Subacute sclerosing panencephalitis is a long-term effect, which we will talk about, bless you, next time. The measles virus causes immunosuppression. It reproduces and kills lymphoid cells, right? Lymphocytes, the cells of the immune system. And that's the main cause of death in third world children. They get secondary bacterial infection. And as we talked about a couple of sessions ago, it erases immune memory. So for some time, until you restore that immune memory, you are susceptible to all kinds of other infections. This is no trivial infection, okay? Do not let anyone tell you that. It's not trivial. How do you prevent it? Before we had a vaccine in the US, there were 3 to 4 million cases of measles per year, 400 to 500 deaths, 48,000 hospitalizations, and 1,000 kids with chronic disability from encephalitis. Tell me that this is a mild disease. It's a lie to say that. But we introduced the vaccine in the 60s, and we stopped endemic transmission by 2000. It's called the measles, mumps, rubella vaccine, 3 infectious attenuated viruses. We'll talk about those in the vaccine lecture. Unfortunately, in 1998, a gastroenterologist in the UK by the name of Andrew Wakefield published a paper saying that MMR caused autism. The paper was wrong. It was a fraud. It was eventually retracted. Since then, over a dozen studies have been done in different countries showing no connection between MMR vaccine and autism. Yet people still listen to Andrew Wakefield, who's now living in Austin, Texas, purveying this anti-vax nonsense, and they don't immunize their kids. And that's why we have outbreaks throughout the US now. And I don't know if you've heard this, the head of HHS, RFK Jr., wants to commission another study to look at the connection between MMR and autism. This is a waste of money. Plus, he's commissioned someone who's an anti-vaxxer. So it's going to be a bogus study. He's not a scientist. He's not a physician scientist. This is incredibly bad, folks. You know, I know you have your thing to do here, but this is really bad. And so now we have outbreaks again in the US. I'll show you. So these were the cases in the 60s, and the vaccine was licensed in 63. A second dose, that brought down the number of cases, as you can see. In 89, we recommended a second dose, and then in 2000, we eliminated circulation. There was just a case here and there, no more outbreaks. But unfortunately, around that time, Wakefield started to do his stuff, and we have cases coming back. So here is measles cases as of March this year. So you can see, boom, 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 various cases. It was a famous one in Disneyland in California, where parents brought their kids, all of them who weren't vaccinating their kids. So there was an outbreak there. This one is an outbreak in Brooklyn. And now this is the Texas outbreak in 2025. And this is, on the bottom here is 2023. So that's 2000 through 2025. And this is a little more granular view, 2023 to 2024. So we have cases. Now these are few cases. But you can see in some years we have over 1,000 cases. We have over 400 so far this year, which exceeds last year. And I'm sure we're going to go over 1,000 cases, and we're going to have multiple kids die. This is totally unacceptable, folks. Globally, there's a ton of measles. Anyway, this is Brooklyn, 2018-19, an outbreak. These are dates, onset of rash. The red is Brooklyn, the Williamsburg. It's an acidic community which did not immunize very well, so big outbreak there. This is the outbreak in the US currently. 483 cases, mostly 5 to 19 years of age, and 97% unvaccinated. We've had 2 deaths, and we've had 70 hospitalizations. Again, most of them 5 to 19 years of age. But in the rest of the world, tons of measles, 663,000 cases in 2023, many, many deaths. These are the different Europe WHO regions, you know, African, forgot AMR, Americas, Eastern Mediterranean, European, Southeast Asia, and Western Pacific. Yeah, I got them all. I can't name all the states probably. So you can see peaks of measles every year, big one in 2019. This is mainly due to lack of vaccination, and this is going to get worse because we used to contribute a lot of money to vaccinating through US aid. That program is gone. Many, many fewer kids are going to get vaccinated. More kids will be dying. And of course, COVID also delayed vaccination programs. These are the countries that have a lot of measles. You can see the top one here is Yemen, Azerbaijan, Kazakhstan, India, Iraq, Kyrgyzstan, it's Russia, a supposedly civilized country which spends money on oligarchs rather than people's health. And this is why we have so many cases. But we're going to get there too, because we're going the same route, folks. I want to get you upset about this because this is serious. These cases are going to go up, and there's going to be more than just measles, folks, because people are going to stop vaccinating against all things. So the next question is, which of the following is a good reason to get measles vaccine? A 1 in 1,000 chance of getting acute post-infection encephalitis, a 1 in 1,000 chance of death. Each infected person spreads measles to 15 others. Immunosuppression can lead to secondary infections. All of the above. So in Texas, in the Mennonite community, which, they don't do vaccination, they know not to bring their kids to the hospital if they have measles, because you're going to infect so many other people if you go to a hospital. So if you see that rash, keep them home and take care of them. And they only go to the hospital if they have pneumonia and they can't breathe. All right, let's see what we did. All right, all of the above. These are all a good reason to get measles vaccine. Poliovirus. So there are going to be outbreaks of polio in this country too. You will see, right? Racaniello said it. This is an icosahedral virus with a plus-stranded RNA genome. Very stable because it's a nice shell, and that's why it can pass through your intestinal tract. The way you get polio is by ingesting the virus, fecally contaminated material. People have lousy hand hygiene, and they go to the bathroom, they don't wash their hands, and they transmit viruses to other people that way. Virus goes through your stomach, low pH, no problem. Intestine, high pH, bile salts, proteases, no problem. It goes to your intestine, reproduces in the mucosal surface, goes through the lymph nodes, and starts a viremia. And that's it in most cases. You shed a lot of virus, and that's how you could pass it on to someone else. But in 99% of infections, you have a transient fever, muscle ache, and that's it. 1% of people, the virus enters the muscle, goes to the central nervous system, and destroys the neurons, and that causes the paralysis. But only 1% of people. Yet many, many kids were paralyzed in the US, and that made us develop vaccines. And so we made a transgenic mouse model to study polio, as I told you before. And this is a section of spinal cord of a mouse that had been paralyzed after infection. And you can see the motor neurons are all infected, and those are eventually going to be destroyed, and that will give you the paralysis. Now here's the pathogenesis of polio. In the old days, before computers, we drew figures by hand. Okay, we had these fancy pens and apparatuses, and we'd draw figures. So this is hand-drawn, okay? We also had press-on letters that we would rub. Now you do this on a computer. It's great, but we had to do it by hand. And that's what this is. This is a compilation of symptoms and signs from a bunch of people who developed polio. So let's see here. We have days on the x-axis and different things on the y. So here, the temperature here. So this is exposure here. After about 7 days, that's your incubation period. You don't feel anything. Then you get a headache, sore throat, nausea. You get a spike in fever. And for most people, that's it. It's the end of the infection. They shed virus in the saliva and in the feces for a long time. You can see over a month. There's virus in the blood. But a number of people go on for another 5 days, and they develop more symptoms. The fever spikes again, headache, nausea, stiffness, pain, and paralysis. And that coincides with virus being replicating in the central nervous system here. Again, this is 1% of the people who are infected. We're not sure why only 1% are susceptible to paralysis, but it's probably some single nucleotide polymorphism in one of the immune system genes, or one or more. Now, humans are the only reservoir of polio. Nobody else is infected. They're spread in nature. They're spread by fecal-oral transmission, as I said. In temperate climates, it peaks in the warm months. And there is a complication called post-polio syndrome, where 30 to 40 years after you recover, and many people would recover, you would get paralyzed again. This is about 25 to 40% of the people this would happen to. It's not an infectious process. There's no virus. It seems to be that, as you age, normally you lose motor neurons. And if you've had polio, you don't have a reserve. And so as you age, you get paralysis again. So you could call this long polio, right? This was way before long COVID. Long kinds of infections happen all the time. So here's the epi curve for the US. Remember, at first, I don't know if I told you this, but polio first emerged in the early 1900s, series of outbreaks in the US. And then after the service people came back from World War II and had kids, then a huge spike in polio. This is the post-war baby boomers right here. And because there's so many of them, they increased the numbers. So we developed vaccines, and we introduced one in 1955, one in 1961, and we eliminated polio from the US until 2 summers ago. There was a case in Rockland County, New York, which we'll talk about later. But there are very few cases of paralytic polio. Although the virus is here, it's circulating in people, and it's in the wastewater. Okay, so that's polio. And now we're going to talk about norovirus. I have a fact for you on this slide. It was given to me by a gastroenterologist at Stanford. In any 24-hour period, 200 million people in the world have gastroenteritis, right? Vomiting, diarrhea. The amount of diarrheal water they pass equals the volume of water passing over Victoria Falls in 1 minute. That's 65 million liters per minute going over Victoria Falls. 65 million liters of diarrheal water is passed every minute in the world from people with gastroenteritis. And it's mostly viral. That's a very cool fact, don't you think? So norovirus is a major cause of gastroenteritis. In fact, this is a pie chart of causes of gastroenteritis. Norovirus, 58% of all cases. And then we have Salmonella, Clostridium, Campylobacter, Staph aureus, and others as well. But norovirus is the queen, and it is a caliciviridae. It's a icosahedral virus, much like poliovirus, with a plus-stranded RNA genome. And causes 50% of all foodborne outbreaks of gastroenteritis. 48 million per year of outbreaks caused by people eating contaminated food. You really need to be careful, unless you want to have gastroenteritis for a couple of days. If you're okay with that, go ahead, eat what you want. It's very contagious. 18 particles are enough to infect you, okay? They've done the study because you can do challenge studies with people. It's the only problem with norovirus is that you lose fluid. So as long as you hydrate yourself, you'll be okay. So in challenge studies, they make sure people are hydrated. 18 particles. I tell you about the medical student challenge studies. If I did, you're probably not going to remember. So at WashU in St. Louis, they have a norovirus group. They take their medical students on Friday. They feed them norovirus. Where do you get norovirus? Diarrhea. They take diarrhea and filter it, add a little strawberry flavoring, and they feed it to the medical students. For the next 2 days, they are vomiting and diarrhea. Monday, they're fine, and they go back to class, they get $300. So when you go to medical school, you think about whether you want to do that. Norovirus is what I call a two-bucket disease, because you're vomiting and you have diarrhea. You need two separate buckets, okay? Fecal-oral spread, as the virus retains infectivity as it passes through the stomach and the intestine. And the virus causes blunting of the villi. Here's the normal villi of the small intestine here. And after norovirus infection, they're blunted. Presumably this has some role in causing diarrhea, although we don't know the exact basis for vomiting and diarrhea. But obviously when you're vomiting and having a lot of diarrhea, you can easily contaminate other people. So fecal oral transmission, actually if you vomit, you could transmit it to people. I'll show you an example with that. Food contamination, water contamination. Oysters, they filter feed, right? And they pull in norovirus. And if you eat them raw, you're going to get a nice dose of norovirus. So I don't eat raw oysters anymore, which is too bad because they're really good. And raspberries, really hard to wash properly because of all those, crinkles on the surface. Often, food handlers are responsible because the virus causes 30% asymptomatic infections. And these individuals can shed virus for a long time so they can go to their job and prepare food, and it's contaminated with norovirus. And that could be food, institutional food, or food at a catering event, or even in a restaurant, whatever. Norovirus is why in every restroom of a restaurant there's a sign that says employees must wash their hands before going back to work. You've seen those, right? That's because of norovirus. Otherwise, they would use the toilet and not wash their hands and have norovirus on their hands. Sudden onset of vomiting, diarrhea, stomach pain, 28 to 60 hours, maybe longer if you have some illness, and 30% asymptomatic infections. This is a problem. That's how it's spread in many cases. The viral shedding peaks 1 to 3 days, but it can persist for 56 days. And the thing is, you can get reinfected. You're only immune for a short period of time after infection, and you get reinfected over and over and over again. We are trying to make a vaccine that will hopefully get around this. Humans are the main reservoir for noroviruses, but I don't know if you remember the big whale I showed you in the first lecture, and I told you that whale excretes 10 to the 13 caliciviruses a day. Well, a norovirus is a member of the calicivirus family, so we think that maybe some of these marine mammals can spread infection to humans. It affects people of all ages, year-round, although there's a peak in cold weather, but you can get it any time. I think we recently had a norovirus outbreak this winter. I was hearing a lot about it. And the outbreaks often occur in semi-closed environments, nursing homes, hospitals, cruise ships, military, schools, recreational activities, anything that favors person-to-person spread. Now, it wouldn't happen in this setting because I'm up here and you're not touching my hands, so I'm not going to transmit it to you. So the only thing you can do right now is to give people fluids because they're losing a lot of fluids, right? How many million gallons a day? So give fluids. But, in countries that don't readily have fluids, so that's a problem. And so young kids can die. 200,000 deaths a year globally, mainly in children who cannot get proper hydration. And I think this will go up again because of the end of U.S. aid. So here are some pie charts to show you how it moves. These are settings. So the biggest culprit here is restaurants and catered meals. So the preparation people are contaminating the food. Retirement centers, nursing homes, hospitals, schools, daycare centers. And, vacation settings, cruise ships, only 10%. But every time a cruise ship has an outbreak, you certainly hear about it. But it's really only 10%. And the modes of transmission, foodborne 10%, and person-to-person 88%. That's very interesting that mostly it goes person-to-person. So here's an example of transmission by vomiting. This is old, but it's still relevant. An Air New Zealand flight, somebody threw up, right? Happens a lot on planes. And they made a mess, so the crew cleaned it up. The crew got sick, and then on every flight they were on, subsequently, someone else got sick. So just goes to show you how transmissible this is. Next time you hear someone vomiting, you're going to be worried, right? And then there's the cruise ships. You know, Disney calls a cruise ship the happiest place on earth. I don't think so. I actually have never been on a cruise ship, and I never will set foot on one. A big tin can with 5,000 to 6,000 people, all eating food prepared by the same people. No, there's just no way. Anyway, these are outbreaks of norovirus and others. So other viruses can cause gastroenteritis. You can see the unknown here, but the norovirus one are all marked here. And when there's an outbreak on a ship, this is a big problem because the ship has to go back to port. Everybody has to get off, and they have to sterilize the whole ship, which is mostly metal, but still, it's costly for the ship company. So why are they associated with cruise ships? It's only 10%, but health officials track illnesses on cruise ships because there's a lot of people in one place. Remember the Diamond Princess? Yeah, that cruise ship off of Japan that got COVID, and they didn't know what to do with people. They just left them on the ship, and they all got infected. So when you get an outbreak on a cruise ship, you have to handle it quickly. Close living quarters, there's always somebody around you, right? And you're eating food prepared by someone who's shedding virus. And then maybe passengers too are bringing the virus on board as well. So that's why I would never take a cruise. But maybe you don't mind throwing up for 2-3 days and having diarrhea. So how do you protect yourself? Wash your hands a lot. Rinse your fruits and vegetables. If you buy a bag of lettuce and it says pre-washed, just wash it, please. I have seen bags of lettuce with dirt in them that say pre-washed, ready to eat. Don't do it. Cook your shellfish thoroughly. Clean your surfaces, kitchen surfaces, wash your laundry. If you're sick, don't make food for other people. And in my view, don't eat the mince. So in the old days, there used to be mince in a bowl outside the restaurant, next to the cash register of the restaurant. People would just take them as if they didn't have enough to eat. I don't get it. And those things are contaminated with fecal coliforms. They've done studies showing high contamination with E. coli, which is a marker for fecal contamination. So people go to the bathroom after they eat, they don't wash their hands, and they take a mint. So don't eat the mints. Even if they're in wrappers, don't eat the mints because the wrappers are contaminated. And if you're sitting at a bar and there's a bowl of peanuts, don't eat them unless you see the bartender pour it from the jar into the bowl, because somebody could have just, I see people grabbing food all the time, and that's the way you get norovirus. Very simple to avoid it. Okay, we'll end up with West Nile virus. Now, West Nile virus was not isolated in Egypt, as this cartoon says. We're pretty sure it's West Nile virus because this patient looks like an Egyptian. But no, it was not identified in West Nile. It was identified in the West Nile district of Uganda, which is down in Africa there, 1937. And it was absent from the Western Hemisphere until 1999, which you may remember I talked about this. It entered New York in the summer of 99, now has spread throughout the Americas since then. And we think this virus came from Israel because the sequence is identical to that of a West Nile virus found in an Israeli goose from a goose farm. I don't know how it got here. It infects many birds, many mosquitoes can carry it, other vertebrates can carry it as well. So here is the infection cycle of West Nile virus. So the primary transmission cycle is among birds. The mosquitoes bite birds in the wild and transfer the virus from bird to bird. And wild birds are okay for the most part if they have seen it. When it first came into the US, all the crows and the blue jays were dying because they'd never seen West Nile before. But what wild birds in other countries that have seen it were fine. It also started killing the exotic birds at the Bronx Zoo because none of them had ever seen West Nile virus. Now, from time to time, the mosquito bites either a human or a horse. And that's when we get West Nile fever and the horse gets a similar disease. And that's a dead end. It doesn't go to anyone else. So every human infection is a spillover from mosquitoes, from birds via mosquitoes to humans, right? No human-to-human spread. The viremia is not high enough for a mosquito to be able to take a blood meal and spread it to someone else, okay? And the name for that is zoonotic transmission. We're going to talk about that later. From animal to human, every West Nile infection is a zoonotic infection. The mosquitoes are typically culicine mosquitoes, which are widespread throughout the US. 3 to 14 day incubation period. 20 to 30% of people develop a flu-like illness called West Nile fever. 80% of people have no symptoms. There's no reason to talk about shedding here because we don't shed and mosquitoes don't pick it up from us and spread it to anyone else. 20 to 30% of people, flu-like illness, fever, malaise, etc. But 1 out of 150 develop neuroinvasive disease. The virus gets into your central nervous system like polio and destroys neurons. You have headache, eye problems, muscle weakness, cognitive impairment, polio-like paralysis, 10% mortality, and over 50% of these people develop long-term neurological sequelae. This is no laughing matter, West Nile virus, okay? So you really need to prevent yourself from getting bitten by mosquitoes because this is prevalent at certain times in the US. So the pathogenesis in people, right, were bitten by a mosquito who picked up the virus from a bird. So it injects it into our blood, so it's immediately picked up by dendritic cells, the sentinels. What do dendritic cells do? They bring it to the lymph node, and the virus actually reproduces in lymphocytes. So you make a lot more viremia, goes to the spleen and other lymphoid organs where it can reproduce more. And in a certain number of people, it gets across the blood-brain barrier where it can reproduce in neurons. Now, interestingly, the viral RNA is sensed by toll-like receptor 3, which ends up making TNF-alpha. And TNF-alpha loosens up the blood-brain barrier so the virus can get in. T cells in the brain and interferon are important for protection of the brain against infection. These are the numbers in the United States. So the top chart, we have from 99, which is the first year it was here, and you can see different numbers in different years. It's broken up into neuroinvasive disease, the light purple, non-neuroinvasive, which is purple, and deaths, which is orange here. And so that only goes to 2019. From 99 to 2023, we've had a total, in the US, we've had a total of 60,000 cases, 27,000 hospitalizations, and 3,000 deaths. So as I said, this isn't a trivial disease, and a vaccine is in the making. In fact, it can make horses very sick. So there's actually a horse vaccine because horses are expensive, and especially racehorses are very expensive. So you want to protect your racehorse from West Nile, so there's a vaccine for that. This is an example of neuroinvasive disease by state. It really goes along with the incidence of infection, but basically in the long run, every state has had neuroinvasive disease from 99 to the present. This is number of cases by week of illness. So this shows you the seasonality, right? Number of cases, y-axis, week of onset, July, August, September, October. This is the mosquito season, and that's where most of the cases occur. So you can prepare yourself. And the neuroinvasive disease by age groups, you can see it goes up in years. And so your incidence per 100,000 is maximal in over 70. I don't know if you remember, but a couple of months ago, Tony Fauci got West Nile neuroinvasive disease. And he wrote an op-ed somewhere saying, oh, it was the worst I ever felt in my life. We really need to have a West Nile vaccine. When Tony Fauci was head of NIAID, he canceled the West Nile vaccine program. So he said there weren't enough cases. But there sure are enough cases. So you shouldn't complain, I guess. I like Tony Fauci, but I think he was wrong on that one. So how do you prevent it? Well, culicine mosquitoes bite from the evening to the morning. So if you're planning on going out, dress properly. You use repellents, proper clothing. Your house should have screens on it. And as I said, we have a human vaccine in development, and a horse vaccine is available. There's a 30% fatality in horses. If you have a racehorse, you need to vaccinate it. All right, I want to end with this slide, talking about viruses and the central nervous system. I've told you today about several viruses that get into the CNS. Poliovirus, Measles virus, West Nile virus. These are effectively transmitted by shedding elsewhere. So poliovirus is shed from the gut, measles is shed from the respiratory tract, West Nile is picked up by mosquitoes. So going into the CNS is a dead end because viruses cannot get out. So there's no evolutionary advantage for a virus to go into the CNS because it cannot transmit to another human. It transmits very, very well by other ways, all right? So I want you to remember that. In general, viral CNS invasion in humans is a dead end. Now, in animals, it might not be because animals eat each other, so they could eat a virus that's in the brain and that would be transmitted to another animals. But we don't do that, so there's no selective force to get a virus in the brain. It's an accident, basically. And that's why it's so rare in all of these infections. All right, so that's acute infections. Next time, we're going to talk about persistent infections, the ones that last your lifetime.